You're going to draw three simple labeled pictures and send photos of those three labeled pictures to me. I call this assignment Wisconsin to Boston because it follows the path of our textbook author when he was your age in the early 1970s, and how his research path occurred. You don't have to remember this path at all. But they'll see that he shifts gears when he goes from Wisconsin to Boston, and it's really important for us to understand it. So, it's two physical places, different times. So, Arthur grew up and went to college. He went to graduate school in Florida, and then, nice sunny Florida, and he decided to go to cold, snowy Wisconsin, Madison, Wisconsin, where there was some research being done there. So he went there and worked there for a while doing research, and we'll talk about that research, and it'll be the basis for our first two drawings. And then he went to Wisconsin, and went from Wisconsin to Boston, and he shifted gears. He studied this same gene, but he shifted gears how he studied it when he went to Boston, and he spent the rest of his career at Boston, at Harvard, and Boston University. So again, we don't need to remember the details of that. Just, I'm going to emphasize, he moves, and he studies the same gene in a different location using a very different strategy, and it's really important for us to understand it. So here's the guy, and again, you don't need to remember this guy's name. Here's the scientist that was working in Wisconsin, and he's in our textbook in a page because he's famous for studying what he studied in Wisconsin. So our textbook author was excited to go to the Wisconsin and study in the snow in this guy's lab. And what this guy was studying was the replication, so how a certain kind of virus reproduces that's called a retrovirus. So there's a certain category of viruses, of which there are a bunch of different kinds. One example modern people have heard of is HIV. So a human immunodeficiency virus, and it's a retrovirus. So it just has to do with sort of the process of the virus infecting cells and replicating and leaving this infected cell. And so here's a picture of this, and this is going to be the basis for our first drawing, and it's the replication of any retrovirus. And I'll just talk through it here, and then we'll draw it. But it's this drawing, and your drawing doesn't need to look as detailed as this because, for example, the first thing is just it's going to infect the cell. And what is infecting the cell? This virus. And the virus has these black portions here. Those are proteins that protect the virus when it's out in the environment, and they also help the virus infect the cell. And then inside the virus is the genome, the genes that the retrovirus needs to go through its replication cycle. And these genes, in this case, are RNA. And so retroviruses are kind of RNA virus. So when the virus is out in the environment, it's a protein coat with RNA genome inside. And that's not like all viruses. There are many viruses that are DNA. Then of RNA viruses, this virus is a special category of virus called a retrovirus. So that's why it's called a retrovirus. Here's the first step of why it's called a retrovirus. When the RNA of a retrovirus comes into the cell during infection, the first thing it's done is what's called reverse transcription. The RNA is going to get copied into DNA. And we're used to gene expression that we've been learning about in our course, where the DNA, the gene, is transcribed into RNA, right? And so that's considered the normal pathway. And this is the opposite. Here, the RNA is introduced to the cell, and it's the viral RNA, and it first gets transcribed into DNA. And because that's the opposite direction of what's usually done, it's called reverse transcription. And you see I put an arrow here because this is going to be the basis for our second drawing, is to learn more about this step, this reverse transcription step. And so our first drawing is going to be this life cycle. Our second drawing is going to be this. And our third drawing is going to be something about this. So that's why I put two arrows here. The first one is the retrovirus life cycle. We're drawing it right now, and we'll get to the second and third one. And they come from this pathway that we're talking about. So just to mention a little bit about this, there's an enzyme that does this special process. So an enzyme grabs here and copies the RNA into DNA, and that enzyme is one we want to know the name of, and it's called reverse transcriptase. And it's not shown in this life cycle, but this RNA virus has to have a gene, and it functions like RNA. It gets translated by the ribosome, so the ribosome is going to grab that upon infection, and it's going to synthesize the protein, reverse transcriptase. So that's a protein that the virus needs to provide upon infection. So we're going to copy it into DNA. It gets turned into double-stranded DNA, and then the next step in the the replication cycle is it gets inserted into the chromosome, into the host DNA. So that's DNA, and we're in the nucleus, and this would be part of a chromosome. And so the part of the replication cycle is just upon infection, it gets reverse transcribed into DNA, turned into double-stranded DNA, and then inserted into that chromosome, into the host DNA, and it gets inserted at a random location. And then what happens is now our regular RNA polymerase, like our gene expression RNA polymerase, starts to transcribe that. So now it's just normal transcription, and now the virus doesn't need to provide that enzyme. The RNA polymerase is already there. So now the process of replication is let's copy a bunch of the RNA, and then let's assemble it into the protein coat, and it can leave the cell to go on to infect other cells. And so a key concept is that through this replication process, we're first going to use a enzyme provided by the virus to reverse to transcribe the RNA into DNA. Then it's going to get inserted, and then we're going to use the host's own enzymes to transcribe that inserted DNA into RNA and package that RNA and have it leave the infected cell. So that's our first drawing. And I'll just draw it so you can see how simple our drawing is going to be. So here's a cell, and here's a virus, and we want to make the protein coat. And we want to make the RNA genome, viral genome. So that's going to infect the cell, and the protein coat is going to be opened, and the RNA is going to get into the cell. Then the RNA is going to have the enzyme reverse transcriptase bind to that RNA, and it's going to reverse transcribe the RNA into DNA. So now that's going to be a DNA, and that is going to get double-stranded, and then it's going to go into the nucleus. Where it's going to randomly insert into, maybe we'll make these a little different color, it'll randomly insert into the host's DNA, which is in the part of the chromosome. That's what chromosomes are made of. And so we'll get that inserted. And it's going to get inserted and inserted randomly. Okay? So then the next step is that our regular familiar RNA polymerase is going to bind there like it does, and RNA polymerase is going to transcribe in a normal way this viral genome, and it's going to transcribe it into new copies. And those new copies of RNA will be, I should have written them by maybe the same purple color because this is the new viral genome, but maybe it's good to have it be a separate color. And lots of these get made, and they get packaged into the protein and leave the cell. You'll make some more of these as a new replicated virus. And that's it. That's our first drawing. Okay? All right, great. So to make that drawing, label it, understand what's going on in those steps, especially appreciate this step and this step. And we'll move on, okay? So then the next thing is that our author moved there, and our author was really interested in studying that topic. This reverse transcriptase enzyme went on to be used in experiments. And so it became a really useful enzyme, and so the scientists that were working on the enzyme in Wisconsin won a Nobel Prize. And so this goes back to this, which is discoveries getting turned into new tools. And we've learned about an example of that in class, and we'll learn about more. And it's no big deal. It just happens a lot, and it's interesting to see when it does. So now we're going to describe how it's used as a tool. So it's an example of a neat trick, and it's the RNA, the reverse transcriptase from that, the RNA retrovirus is actually used now to study mRNA. So it's not used as an infection. The scientists just take that one enzyme and they use it to study mRNA. And what they study is they study something called RNA splicing. So let me draw that, and now we'll draw our second picture. And we might have to draw two pictures just to show it, but here we go. Okay, so it's going to look like this, where we start out drawing, and here's our gene, here's our DNA, and here's any gene. And this time we're going to make the transcription unit really long. So there's the transcription unit, there's the promoter, there's the enhancer. So this is the transcription unit. Ooh, spelled that, messed up. And then also we're going to have, as part of the DNA, so I'll make all this brown, this is going to be a coding gene, and so that's the coding region. The coding region is a subset of the transcription unit. And so this is going to go on and get translated. So now we haven't talked about this yet in our class, but to start out with, this is what gets transcribed. So RNA polymerase grabs here and moves down the gene transcription unit, copying the DNA into RNA. And this is true, like we always talked about. This, the first RNA, there's RNA that gets made, is exactly the same length as the transcription unit. It defines the transcription unit. They're one and the same length. It's just that the DNA gets transcribed into RNA. And this RNA has a special name, and it's called the primary transcript. So it's just the first name, first RNA that gets made. Now, in the nucleus, before the RNA leaves the nucleus, it's going to go through here. So now we can make like several RNA molecules like this. Oh, no, sorry, I should have made them purple, sorry. I'll draw that over. So we'll get multiple RNA molecules like that. And as we go copy that RNA will get ready and it will leave the nucleus. And it's going to be a coding gene, it's going to be coding, it's going to be mRNA, and it's going to leave the nucleus. And it starts out as the primary transcript, and when it leaves the nucleus, it's called mature RNA. And in between, it then goes several steps that are called RNA processing. And the one we want to draw about here is called RNA splicing. And we haven't talked about this in our class, but when we look at the primary transcript and the mature transcript, so the mature mRNA transcript, the mature transcript is often shorter than the primary transcript. And that's because some of the primary transcript has been removed. It's been spliced out. And we can just draw a picture of what that looks like. So in the primary transcript and also the DNA, so I'll draw the boxes like this, there are parts of this that are going to be retained in the mature mRNA. And then, surprisingly, there are parts that are not going to be maintained, and they're in both the DNA and the primary transcript, but that portion gets removed and deleted. And the entire gene looks like that, where there'll be portions, and they could be short or they could be long, and there can be several of these, and each gene is different. Some don't have any of these, some have many of these, 30 or 40 of these. And so in the primary transcript and in the DNA, you can see there are portions that get removed before the RNAs mature. So these are essentially garbage that gets destroyed. And those portions are called introns. And so the DNA has introns in where? In the transcription unit. The primary transcript has introns in the primary transcript. The mature RNA has no introns. That's why it's shorter. The introns were deleted and destroyed. These exons are all present. So we see 4 of these down here, and these are called the exons. So exons and introns. Exons are saved, introns are destroyed, and the exons just join together like that, directly together, to make the mature mRNA. So the mature mRNA that leaves the nucleus is composed of exons, then no introns. So again, introns in the DNA, the primary transcript, but not the mature RNA, exons in all 3. There are exons in each one of these portions. They don't get removed. And so as a result, the DNA in the transcription unit is kind of frustrating because it includes lots of portions that are garbage. And so the next part, and we might need to make a little second drawing that I'll try to show you, is simply this. Scientists, maybe I'll just draw it over here. So scientists can take the mature mRNA and they can study that. But it's hard to study the RNA. And these are exons. Oh, and I mentioned I think I was going to number them. So people just number the exons like that. 1, 2, 3, 4. So they are 1, 2, 3, 4. And people really want to study the mature RNA because that's the functional protein. Like, for example, look at this. This is maybe where the exons include portions of the coding region, we could say. And you could see how now the coding region is all together, but in the original DNA, the genomic DNA, the coding region was split up. And so that's really frustrating to study. And so what was done is companies took that enzyme, reverse transcriptase, and they took the gene from the retrovirus that codes for reverse transcriptase, and they put that gene into bacteria so they could grow the bacteria and harvest the reverse transcriptase protein, and they could sell it. They could sell it because scientists could purify this mature RNA that's out in the cytoplasm, and they can use the mature RNA and the reverse transcriptase to copy the mature mRNA into DNA. And it's a lot easier to study DNA than RNA, so they would do that, and then they could study the DNA, and the DNA would have no introns. And so this is our second drawing, is to draw first the concept of intron exons and the primary transcript having introns and the mature mRNA having no introns. And then over here, drawing that we could take all the mature RNA, copy them into DNA using reverse transcriptase, and it would have no introns. So we wouldn't have to study them at all. And so it's just a neat trick to allow us to study any genes in any animals or plants. No introns. And because this is not really, it's natural DNA, it has the AGCT nucleotides and standard biochemical structure of DNA, but it's not a DNA that's found in nature because it doesn't have any introns. DNA found in nature typically has introns in the transcription unit. So this is a little bit different, and so people wanted to give this a special name, and it's called cDNA. and it stands for complementary DNA. It's complementary to the mature RNA. And so that's our second drawing. Is we're going to use this as a trick or a tool after it was discovered in Wisconsin. So I'll just go back to this and show you that it's an example of a new trick. Here's an example just from the web of how the mature RNA gets spliced together in the nucleus to make the primary transcript gets spliced together to make the mature transcript. So that's part of the process of this RNA processing. And there are other steps that occur in the nucleus to make the RNA ready to be the mature RNA that is going to leave the nucleus. Great, okay? Then the next thing is we're going to focus on this. So let's see if I can get to a map. No, there's our author. And our author went to the Wisconsin lab because one of the projects that the lab was doing was very interesting to the author. And so let's just draw this again. So the author's down here in Florida. And this lab in Wisconsin was famous for studying the retrovirus replication cycle. And the scientists were making discoveries that were great value. They went on to use reverse transcriptase, and use it as a neat trick to study cDNA and mature mRNA that codes for proteins and so forth. And so it's an exciting time in this lab. And you could just say one of the projects was to study the retroviral replication cycle. Okay, so they had a variety of retroviruses that they were studying in the lab, right? Because that's what you want to do. You want to study them to see how do they all replicate, and they all replicate that same way. So these can be, I mentioned HIV is in humans, but there weren't at this time, when the work was being done here, there weren't any retroviruses known in humans. So these were done in different species, like there was one in a cat, and there's a monkey, and so forth. There's a rat, chicken, and just retroviruses that were out in nature. And our author came to this lab studying all this stuff for a specific reason, and that is that some scientists had discovered a rat retrovirus. So it wouldn't hurt us. It was just a virus in rats. And this rat retrovirus, if you infected it into a new rat, it would cause cancer. And it immediately caused cancer. The cells would become cancerous, and it was a specific type of cancer. It was called a sarcoma. And we'll learn about sarcomas later in our course, but it's a very specific category of cancer. It's really important to understand that there was a retrovirus that just caused disease, not cancer, and then there was a change, a mutation, where this virus could cause cancer. And this will be the basis for our third drawing, is our textbook author, Jeffrey Cooper, wanted to go study this and learn about this retrovirus. Because it could cause cancer, this mutation made this what was just a disease retrovirus. It made it be a cancer-causing retrovirus, and this was they called it an oncogenic retrovirus. And oncogenic means cancer-causing. So it's really important to appreciate that there was a mutation that turned this disease-causing retrovirus into a cancer-causing retrovirus. And our author wanted to understand that mutation. So our author was there and studied it, and this is what that work looked like a little bit, just to show you. So he's going to study it, and our author is going to find a oncogene, it's a very famous oncogene called RAS, and it stands for rat sarcoma retrovirus. Again, it was a mutation. The negation of RAS emerged during the extensive study of acutely transforming retroviruses in mice, rats, cats, monkeys, chickens, and turkeys. These oncogenic viruses caused rapid formation of sarcomas in infected animals and potently transform cells in culture. That means make them cancerous. Okay, so our author's career started by studying this oncogene, and he was the first person to really, he and others were the first people to really identify what was going on. And this is what was going on, that there was this problem gene, and the problem gene looked like this. So this is chapter 19, and so we'll get to that chapter. And this is one of the retroviruses, another one of the retroviruses that were studied, that were these mutations that were called oncogenic retrovirus, because the author was studying one from a rat, but others were studying ones from other animal species. And this shows a normal retroviral genome. So that's the RNA that's inside the protein capsule that gets inserted. And so it has some genes here, and these are retroviral genes. The envelope gene makes those black proteins that are the coat, and then this makes the reverse transcriptase. So I mentioned at the start of the video that the cell infects, and it needs to use the RNA to be translated into some proteins. And then this GAG protein is another protein involved in the infection process. So great, that's what the retroviral genome looks like. It has some genes. This is the oncogenic retroviral genome, and it has an extra gene. See, there are all the normal genes here, and there's an extra gene. And this one in the textbook is called SARC, and this is what our author studied that he called RAS. So it had an extra gene, and our author studied that and identified that the oncogenic retroviruses, and he studied the rat one, and across the world there were about 20 or so oncogenic retrovirus mutants of different strains and different species that were being studied. Each one had the same story. It had an extra gene. And so this is our third drawing, is we want to draw the normal retrovirus genome and then the oncogenic retrovirus genome to see that it had this extra gene, and our gene is called RAS. Oh, and here's the protein products down here, okay, for the normal virus retroviral genome genes, and for that RAS, okay? And here's what's happening. So if we go back to this, we want to emphasize that the DNA gets inserted here. And what happened is, during one infection process in one rat, the retrovirus got reverse transcribed, and the double-stranded DNA got inserted, and it happened to get inserted right next to a gene. And the gene was in the host genome. The gene was a rat gene. The gene went on to be called RAS. And so the mutation was that by random chance, an infected rat cell had the retroviral DNA inserted next to RAS, and when that got transcribed, the product included the retroviral genomes GAG, PAL, ENV, plus RAS. So now this has RAS. This did not. This is the cycle of one animal infection where the mutated oncogenic retrovirus was created. It was created by having it accidentally insert near RAS and then include RAS in the retroviral genome. Scientists, other labs found other oncogenic retroviruses that had different host genes. So the excitement became quickly that it's the host gene, the host RAS gene, that's the oncogenic gene. It's not the virus at all. It's that you're bringing this host gene, and there's something wrong with the host gene. And so scientists quickly came up with this name, where they called the normal RAS gene CRAS, for cellular RAS, and they called the oncogenic gene VRAS. And that's the gene that if you put it back in a rat cell, it would cause cancer, even though it was a rat gene. there was some problem with it. And in this case, scientists later determined that there were mutations in the coding region. And when this went on to infect future cells, it would make a mutant RAS protein. So we can just go over to this picture and say that, yep, this is RAS, and the RAS has some mutations in the coding region. Yep, okay. So again, this is important that we're talking about not just retroviruses, but these specific mutants like that. And on the first exam, we will have learned about 2 of them. So RAS is one, and we'll learn what it normally does in the host. And then another one we'll learn about before our first exam is MYC. And so these were all first discovered in the 1970s in a variety of labs, and they were all discovered because they were host genes that had been picked up by a retrovirus for the reason I just described, that the retrovirus accidentally inserted into the host's chromosomal DNA right next to these genes. And then the retrovirus took those genes with it, and it took a defective version in each case. So these different ones were found in rats or cats or mice or chickens or monkeys, not humans. But in every case, there's a human ortholog. They didn't know that yet, but it's true. Okay, so now I find the map. I don't know if I have the map here again, but let's just see. Yes. So our author went from Florida to Wisconsin, where they were studying retroviruses and just the normal replication cycle of the retrovirus. And one of the projects in the lab involved studying the oncogenic mutant. What's going on? And so our author, Jeffrey Cooper, studied that. And he found that gene. And the gene was in rats. Okay? And so he used that, and other scientists used that in the 1970s to get kind of a foot in the door on what an oncogene is. And an oncogene is a normal host gene that could get mutated and contribute to cancer. Now, here's the last thing, and we don't have to draw it, but then our author moves to Wisconsin. And he leaves the retroviruses behind. So no more retroviruses. What he takes with them is the RAS gene and the knowledge that the RAS gene could have a mutation that now causes cancer. So he goes to Wisconsin, and I don't think I have any slides. I do have a couple slides of it, but let's just get to it like this. And yep. So in Boston, he's not studying the retrovirus anymore, and that's why we say, he moves. He's not studying retroviruses, so no more retroviruses. Instead, he knows, there could be a host gene, in his case a rat gene, and it could cause cancer. So here's what he does, is he takes a tumor in Boston, so nothing to do with a retrovirus. He's left that behind, and it's really important to emphasize that he and other scientists started out with viruses. They found these host genes that were defective and causing cancer, and they said, well, if we could get that gene to cause cancer, maybe the tumor already has a mutant RAS, maybe gene, because it doesn't have to have a retrovirus infection. If it just has a mutation in the RAS, maybe that could cause cancer. So that's the idea. And in Boston, what he did is he took a tumor, and he took the DNA from that tumor in pieces, and he put the pieces into a normal mouse, normal rat, and he found pieces that could cause cancer. and he had tagged those pieces that he put in, and when he looked at which gene, he found that it was the RAS gene. So that's the common piece between Wisconsin and Boston, that it's the RAS gene both times. He was looking for it. He was thinking he was going to find it, and he did. And so this shifts us from seeing that, yep, you could have this retroviral mutation that gets you started understanding it. But if you just shift over and you just looked at normal rats or normal humans or normal monkeys, they have those normal oncogenes in them. And if that normal oncogene gets mutated, it can cause cancer. Yep. So let's just look at these last couple of slides. And so we actually have a small gene family in us. We're not going to worry about this at all. It's just RAS. For us, we're just going to refer to it as RAS. But it says together, those three are the most common oncogenes in human cancer. Mutations that permanently activate RAS, and we'll learn how, are found in 20 to 25% of all human tumors and up to 90% in certain specific types of cancer. So it's really super common. And here from chapter 16 that we're going to get to next, I got to start calling it the right chapter. Chapter 16 in our 9th edition textbook. Here is RAS. And I already showed this in our lecture class one time very briefly. So RAS works on a growth factor signaling pathway. Down here is the target. It's ALK. ALK is a transcription factor. And so this growth factor is activating ALK in the same way that a growth factor could inactivate FOXO. Okay, so we're going to activate ALK. And that's all that's protein, it's just not short on here. So that's RAS. And RAS is in a normal growth control pathway. And when mutated, that pathway can be defective. And RAS can just be constantly activating the pathway even though there isn't a growth factor. So we're going to stop there. And again, 3 drawings. The retroviral life cycle, the role of the splicing story, and then the role of how a host gene could get picked up and included in a retrovirus and turn that into an oncogenic retrovirus. Okay? Great. So that's where we're at with that assignment. And just email those 3 pics to me. Great.